All right, welcome back to the last exploration in Unit 5. Today we are going to look at Unit 5, Lesson 2, Exploration 3. And we're going to be revisiting a topic that we touched on briefly last semester, and the topic is cell differentiation. Now, again, we looked at this a little bit when we looked at how the body is organized in the section that we talked about cells and how the structure of a cell is what determines its function. And so we're going to look at that again today, and specifically what role mitosis plays in that process of differentiation. Now, as you know, skin cells can divide and make new skin cells. Single organisms are able to copy themselves directly, but how do we get those specialized cells in the first place? Your body, as you know, begins as a single fertilized egg. We call that structure a zygote. So from one single cell called a zygote, we are able to grow into the trillions of cells that makes up each one of us. And so let's look at that a little bit more specifically. Now again, this process is called cell differentiation, and it's where a group of general cells take on specific structures that allow them to perform specific functions. And that's a key component, because remember, if you remember back to last semester, the way that our cells are actually shaped, their actual physical shape, is very important to the effectiveness of the job that they can do. If you remember, we looked at the difference between a nerve cell and a muscle cell. And we showed you how nerve cells are specifically designed to be able to transmit an electrical signal from one end of the cell to the other, whereas muscle cells are designed to be able to expand and contract because that's what their specific job requires. Now, we're not going to talk a lot about how those cells, as they begin to grow, convert from a zygote to an embryo to those sorts of things, but I do want to touch on some of the key differences between plant cells and animal cells. You know, the idea around this process is that the location of the cells in that clump matters. That cells that are on top versus on the side versus at the bottom are going to develop into specific structures for that organism. So I show you this picture just to show you that the location of those cells in that clump as it expands before it differentiates matters into what those cells will ultimately become. Again, specific information here about how those cells develop from the embryonic stage. We're not going to spend a ton of time looking at that. If you are interested in that, it's a really, really cool study. Embryonic biology is super cool. Lots of really intricate information to be learned there, and you know, if you guys go on to higher levels of biology, you probably get a chance to study that. But for now, we're going to kind of move on. Okay, the piece that I want to spend the rest of our time talking about is stem cells. And again, we referenced this back in the first semester during Unit 1. But stem cells are those cells that can become anything. They are the types of cells that have yet to be given a specific job, and so therefore they can still be turned into any of the number of different cells in your body. Now, stem cells are really important. As those stem cells begin to migrate to the area of the embryo as it develops, they begin to take on kind of a path almost. Like, they can't necessarily become every single cell in the body anymore, but they can still have a couple different options. The adult stem cells that still exist in your body are in that kind of a waiting phase. They're on the highway. They've maybe gotten off on one exit, but they haven't specifically gone to their ending location. They haven't necessarily become exactly what they're going to be. This gives your body the flexibility to meet whatever needs that it has without having to necessarily copy an already existing cell. They can tap into some of the adult stem cells that exist and turn those adult stem cells into something that's needed. Now, like I said, your body has 200 different types of cells. And when you're in the embryonic stage, those cells can literally become anything that your body needs. Now, the process of acquiring stem cells used to be really controversial because it required the termination of an existing pregnancy. But now, as our technology has increased, we've developed ways to harvest those fertilized eggs after an in vitro fertilization. In vitro meaning a process where the egg is fertilized outside of the woman's body. So you take a sample of an egg cell, a sperm cell, you fertilize them together in a petri dish. And this is done for people typically who are trying to get pregnant and are maybe having trouble without the use of technology. And so you can create several of these embryos all at once, and that allows you to then see them grow and even harvest those stem cells from that petri dish. Now, again, embryonic stem cells are super important because they can become anything. And as we look into the application of this, there's some pretty cool ways that those stem cells are being used. We're using them to treat specific medical conditions. Everything from bone marrow replacement to replacing damaged brain tissue to replacing, even like an example of diabetics. They're using stem cells to regrow areas of the pancreas or repair areas of the pancreas so that people with diabetes can produce their own insulin again, which is really cool. The more we study stem cells and the more we are able to apply them to existing medical problems, the more we see how useful they are. There's even some aspects of cancer treatments that are being done with stem cells where areas of the body that have become damaged by a tumor can be removed and regrown with stem cells, and you can inject stem cells into a tumor and those native cells will take over and effectively push out those tumor cells so that they can't function and continue to grow. Really cool stuff in this area. Now, we're not going to talk a lot about gene expression because this is really something more that we'll talk about when we get to DNA. But I did want to introduce it to you because the expression of a gene is what turns a stem cell into a regular specialized cell. Your DNA is full of genes. In other words, segments of DNA. You can think about it kind of like chapters in a book. And each chapter contains information for how a specific part of your body is supposed to function. Now, obviously, not every single cell needs all of the information. The skin cell on the back of your hand doesn't need to know how your liver cells are supposed to function. And although the information is there, it's what we call switched off. The only part of the DNA that is turned on or switched on is the section of DNA that is related to the function of that cell. So when you turn on that specific set of genes for that cell on the back of your hand, then those genes can be expressed or read. The proteins that are needed for that cell can be produced, and as that happens, the cell begins to be changed into the specific shape to perform the specific function that is needed. So gene expression, again, we'll go more into that and how that works later on in our unit about DNA, but I wanted to introduce it to you because I do think it helps to explain how that stem cell turns into a specific specialized cell with a specialized function. And it's through that process of keeping most of the genes switched off and only turning on the genes that are needed to provide the instructions for that specific cell wherever it's located. All right, so that does it for Exploration 3, and that does it for Unit 5.